By understanding what nature does, we can design better ways of dealing with not only medicine, agriculture, and the environment, but also national security.
This is the beginning, not the end.
We're here to discover new things.
From the most humble microorganism all the way through to this remarkable complex structure we call the human body.
To help our human bodies, to help our environment.
To uncover mysteries of the world and use those discoveries to solve real-world problems. I'm Josh LeBaire. I'm the director of the Biodesign Institute, and I'm welcoming you here to our 6th annual Arntzen Grand Challenges lecture, which is named in honor of Charlie Arntzen, who was our first founding director of the Institute, and whose family actually contributed to the creation of this whole lecture series. So this comes a year after, it's hard to believe it's been a year since our 20th anniversary. So the Institute is now 21 years old. Kind of a reminder of kind of where we've come from and what's, how much we've grown since we started. We now have over 170 faculty members, something like 1,500 people in one way or another work in the Institute now. 3 large buildings. It's really a remarkable place to be part of. So, as I thought about what to say to just get this started, I was reminded that in this past year, I was invited to submit an essay for a humanities magazine, which is not something that I have any knowledge of how to do. They gave me 8 months to write it, which in my mind meant that I didn't have to start it for 5 more months, which is a big mistake, because it turned out they wanted 5,000 words, so that was a little surprising. But the question was, looking back from the future, what did science get right in our time? And as I thought about how to answer that question, I realized, first of all, looking at the other people asked to submit, I was the biomedical guy. There were, poets, there were economists, there are all kinds of leaders, people much better at this sort of thing than I am. But I felt like I had to sort of think about what has biomedicine gotten right. And actually, speaking for the biomedical community, I'm quite proud of us. If you think back on it, at the turn of the 20th century, the average lifespan was around 45. If you look at where we are today, it's closer to 80, and probably all of you know somebody who is thriving in their 90s. So nearly doubling of the lifespan. And I would argue that that is due to science. I have no question in my mind that science is what's enabled that accomplishment. And science in many ways, shapes, and forms, but all of them in their way have contributed to this. And so as I tried to think about what could I say was like the key thing we got right, you couldn't think of one, right? I mean, there's been antibiotics, there's been better purified water, there's been, healthcare delivery, there's been, improved surgeries. The list goes on. And then, of course, drug development and all kinds of things, diagnostics. So, but what really came home, as I thought about it the most, is what really changed for medicine was really the adoption of the scientific method. And it's the scientific method that I believe really deserves the credit for where we are. And it sounds simple enough, but it's this idea of taking an observation, making a hypothesis about what it means, testing that hypothesis and getting others to believe it by doing peer review, and then from that, getting a result that you can then build your next observation on. And I thought of it as sort of a ratcheting system, right? Every time you click forward, you have a backstop that prevents you from sliding backwards because you've now established that one fact. And if you do it again, you get that much closer, and each time you take that ratchet, you get closer and closer to the truth. And that's really what all of us do here. That's what all of you do. Every time you publish a paper, every time you defend a thesis, every time you make a discovery, you're adding one more click to that ratchet. You're adding one bit more knowledge that gets us closer to the truth, and that's how we've advanced. What changed in medicine was, which was once a field that really was built on kind of a master-apprentice approach, where doctors would teach young doctors how to do things, and the reason for doing it was because that's how we do it. That changed in the 20th century to an evidence-based approach, which was, let's see what's the best way to do this, and let's measure it and see how it works. So I think, to me, that's the reason we're here. It's the reason for this institute. It's something that we do not just in the context of medicine, but also in the context of sustainability, in the context of security. In any major problem that faces the planet, biodesign was created as an institute to sort of address those problems, recognizing that we take a scientific approach, that we look at nature as our inspiration for how to address that approach, and that we do it from all the angles that are necessary to get the problem solved, because you don't really solve problems from single directions. So I want to just mention a couple of recent successes that we've had, just a few of our centers, and these are just sort of randomly picked from the last few months of publications. So Josh Hyatt's Center for Biosensors and Bioelectronics has developed an electronic way to store information in DNA. That's a really remarkable discovery. Jordan Yaron and Kaushal Rege from the Center for Biomaterials Innovation and Translation identified serpin B3 as a protein that acts as a tumor suppressor and may be useful in cancer therapy. Tim Long, whose Center for Sustainable Macromaterials and Manufacturing, has been working with companies to come up with ways to build polymers like plastics, but which don't persist forever, but can break down in the environment and therefore are much more sustainable for all of us. Shelley Haydel in the Center for Bioelectronics and Biosensors found, and her postdoc found an essential gene in Mycobacterium tuberculosis. This is the bug that causes tuberculosis, which many of you know infects 1/3 of the Earth's population. So 1 out of 3 people on our planet has TB. So they found an essential gene, and perhaps that may be eventually a target for treating that disease. Ramon Velasquez in the Banner Neurodegenerative Disease Research Center is looking at the role of the herbicide glyphosate, which easier to say Roundup, and how that may be leading to dementia. And then, of course, we all have to acknowledge the team from the CXFEL group. They have now assembled an instrument that has already, for the first time this year, solved a protein structure. So we've actually used that X-ray to make a protein structure. So let me stop with talking about the Institute and just take a moment to acknowledge the amazing things that Charlie's done. And I don't know if we have that ready. Why don't we put that up?
I think everyone needs to be shocked with new ideas occasionally. Something that changes your day-to-day thought process, and all of a sudden, bam, that's really exciting. I had no idea that that was happening out there. I was taking a weekend off in Thailand. It was there for some collaborative research. Went to the river market where the boats come in, and I saw a woman with her baby buy a banana, peeled the banana, and gave it to the child. And I thought to myself, what can I do to re-engineer a plant so it has direct medical purpose? In talking with one of the professors at Baylor, we pulled together the idea, let's produce vaccines in plants. When I came to ASU, I was given an endowed professorship funded by Florence Ely Nelson. I plowed that money into the first studies we did of vaccines produced in plants. And it was at a time when the ideas we were generating were crazy enough that it was hard to get funding from the federal government. Few scientists in their career actually see something that started on the bench and goes on to have an impact that's tangible. And you can make a straight line connection between the two things. So the idea of a Grand Challenge lecture series was bring in new ideas, bring in the cutting edge of science, and create the opportunity for our students, the faculty, everybody in biodesign to interact with this, with the ideas and with the people bringing them to the forefront. I'm Charlie Arntzen. Welcome to the Arntzen Grand Challenges lecture series.
Okay, so we have a real treat for you today in our lecture speaker today. So she's a perfect fit for biodesign because she kind of covers everything that we do. Barbara Hahn is a disease ecologist for the Cary Institute of Ecosystem Studies in Millbrook, New York, sort of an independent institute there. She works at the intersection of ecology, computing, and global health. She partners with collaborators all over the world, including IBM, NASA, various universities. She's pioneered the use of machine learning and artificial intelligence tools to understand when and where new zoonotic diseases are likely to emerge, something that's obviously important to all of us in our health. This has impact in surveillance programs and global health preparedness efforts, land management decisions, and a whole wide variety of other things. So we are just really thrilled to have her here today. Anyone who's been lucky enough to meet her today, she's charming, and I'm going to hand you off to Dr. Barbara Hahn.
How do I sound? Is it on? Is it good? Okay, I'm getting oh, that's gonna be hard for me. Yeah, okay. I have a tendency to do the thing where I trail off. So I'm gonna try not to do that today. Okay, thumbs up? Great, well, it is really such an honor to be with you all here tonight. What an amazing place this is. I have felt like from 8 a.m. to now, my schedule has been almost entirely packed. As they do when scientists go to other institutions to give lectures, you often get to meet with as many people as possible. And today was no different. But today was different in the sense that the day just absolutely flew by. I mean, I feel like every half hour I was inspired and amazed and surprised. And then I got to have lunch with students and postdocs, and I felt hopeful for the future. I got to meet people whose work I read for a very long time, that have been part of my journey. And so it is really such an honor to be here among you all and to be a little bit a part of what you guys are doing here in the Biodesign Institute. So I know not all of us in the audience are scientists, so I'm going to try to really explain myself as much as possible. But if you're confused, just give me the confused face, and I will try my best to respond to that. See if this works. So today I want to talk to you about some work we've been pursuing over the last, 15 years. And really, I am passionate about this idea that we can predict the future of infectious disease. And as was mentioned, I am an infectious disease ecologist. And many of you might be wondering, well, what is an ecologist even? What is ecology? And what does disease ecology mean? Ecologists are really interested in studying why there are animals where they are, how many of them are there, why is that? And what's the cycling? How do they live there? What is their interaction with the environment? What controls that? Why does it change? So these questions are very complicated, take a lot of field work, and arguably more difficult to study than things that have physical rules. So I have a little bit of physics envy. I have a little bit of chemistry envy. Because in my world, a lot of those rules apply, but at such a basic level that by the time you're studying the emergence of these properties on the landscape, it's really difficult to pin down why those patterns are emerging. And so let's add on top of that infectious diseases. Infectious diseases make you lethargic. Infectious diseases make you hide. Infectious diseases sometimes don't do anything. Like you don't feel it, you're just gonna go on being your rodent self, burrowing and doing your thing, getting eaten. So diseases have all kinds of impacts on host populations that are often difficult to detect, which is why we need people who are developing cutting-edge diagnostics. They are difficult to sense, which is why we need people who are developing sensors, like and even remote sensing, developing that technology to give us new eyes on the ground. And so that's what disease ecologists try to do. We try to understand what it is about animal species that allow them to become infected, stay infected, transmit that infection. What does that dynamic look like on the landscape? And what happens when you get a transmission event to a person or to another animal? Say it's livestock or say it's your neighbor. How do we predict that happening? And when I first got interested in this work, I came at it from, I was an infectious disease ecologist in amphibians. So I was mostly interested in understanding the wildlife. And mostly I was interested in understanding their behavior. Does being infected change the way you act? And if so, how does that trickle out to affect the whole community? Because as we know, amphibians are often, I think of tadpoles like popcorn, like everything eats them. That's why there's so many of them in an egg mass. And so does it change the behavior of the popcorn, of the tadpole, of the predators that are seeking them, and how does that infection trickle out to affect the whole system? But really in my heart, I was very interested in infectious diseases that impact people. But I knew that I was also very passionate about the animals that carry these diseases. And so that leads us to zoonotic diseases. These are diseases that have an animal host in which they persist all the time. There's a wildlife cycle. And if humans weren't in the picture, they would just be there forever. They would be cycling, doing their thing in these host populations and living their lives. The problem is that zoonotic diseases transmit from animals to people occasionally. They spill over, and they can cause sometimes catastrophic infectious events, such as the COVID-19 pandemic, highly pathogenic avian influenza, Zika virus, and others. So today, I want to share a little bit of the work that we've been doing over the last 15 years to push this prediction frontier. Can we make predictions about things for which we have such little information that prediction feels like it's impossible? And when you run up against something that feels impossible, what data can you draw in together and use tools from machine learning to see patterns that get you to that next hypothesis, that take you a little bit further in your scientific method? What tests do we need to run next? Kind of like that ratchet example that Josh just gave us. How do we get just a little bit closer to making action from these predictions that will improve humanity and improve the way that we live on this planet with all of these organisms? I thought this illustration was lovely and reflective of the art and science integration that I've been seeing all over the Biodesign Institute. This is by an artist and was published in the New York Times a few years ago. So oftentimes what we do as disease ecologists or as epidemiologists is we react in kind of a panic mode. So we often operate in this emergency zone where we are aware of the risk because it's already realized on the landscape. We are worried about an infectious disease that is outpacing our ability to contain it. It is declared a public health emergency of international concern, where it's going to sort of outpace the boundaries, spill over into new countries. And so those are the diseases that take the most money, but cost the most in terms of human losses and economic losses. They are catastrophic. Once we've kind of put out that wildfire, we then move into what's like the warning phase. We have a pathogen that has a name, it has a sequence, there's diagnostics available for it, and we try to like buckle in and figure out like, well, what happened here? How can we prevent this from happening? What is the vaccine we need? And some people with a lot of foresight develop these vaccines for pathogens that haven't caused these massive outbreaks here yet, even if there might not be any money in them. And of course, I'm talking about the ZMapp vaccine for Ebola virus, which Charlie Arntzen was the founder and inventor. So I want to push this prediction effort all the way up to the watch phase. How far upstream can we go before something emerges from a landscape that is sort of spillover ready? What does something look like that's spillover ready? Which animal should we be paying attention to? And of course, this is kind of difficult to do because prediction is only really possible when these three things are perfectly overlapping and you can get at that star. So when you want to predict a disease event, you got to know kind of what animal it's in normally, which pathogen you're looking for that will break out, and where to look for it. And if you don't have these things sort of perfectly aligned, you have very little chance of being able to predict a disease event and then put it out in advance. But of course, we don't have a perfect view of the world, and actually, this extra circle that's around our sphere of knowledge, it just goes on forever, right? So that external sphere, it just goes on forever, and the zones of unknowns that are overlapping here, represented by the red stars, they are in this sort of infinite space. And so how do we make predictions when we lack some basic information? So let's make this a little bit more concrete, and I'll give you some examples. So if we have a location, for example, we're very interested in, like the Phoenix area, or a location that is known for having previous spillover events of zoonotic diseases that we care about, we may focus our efforts at discovering new hosts that are carrying pathogens in that location, or new pathogens. So those are two unknowns that we don't have, but at least we have a location so we can double down on our surveillance right there. Okay, so that's one example of two unknowns. Another example, I took this picture with Arizona in mind. We actually have one of the world's experts on rodents, Nate Upham, who's here, who was just on live TV, as I understand, in an interview for his work. But these rodents carry many, many zoonotic pathogens that are very understudied. And in fact, I would argue that if you continue to survey just the rodents in the Phoenix, Arizona area, you will continue to find new pathogens, and you may not actually hit a limit. They may not all be infectious to us, but you will never run out of pathogens that you are finding in these creatures. This one here, I believe, carries hantavirus, Borrelia, which causes Lyme disease, whitewater arroyovirus, which is an arenavirus, and several others that I'm forgetting, but I think that's enough for now. I will encourage you to look it up. So the last one that I'm going to focus a little bit more time on today to give you examples is when we have a known pathogen, but we don't know where it's going to break out next, and we don't know what animal actually carries it in the wild. What do we do in those situations? So I'm going to share two examples from our recent work on this topic. Some of it's old, but I'm going to use filoviruses, which include things like Ebola, Marburg, Sudan virus, these things that you've probably heard of in the news. Actually, we have a new outbreak of Marburg virus that was just announced 2 or 3 days ago by the WHO in Ethiopia. And I will also share some work that we did using machine learning. Well, both of them use machine learning, but this one is focused on SARS-CoV-2, and it was very much responsive to the pandemic. Okay, so this is an illustration that was provided by WHO, and it just illustrates the timeline on which the 2014-2015 Ebola outbreak played out. So you can see the numbers of human losses there, but I think it's also important to remember that there were probably like 30,000 orphaned children as a result of these people dying. There was economic losses that these countries still have not recovered from. And so these are devastating infectious diseases, and it's not that they're breaking out because we haven't been trying to find the reservoirs. We've been looking for the reservoir in the wild since the 70s, and they've literally captured tens of thousands of animals. They've sampled plants to try to figure out what it's coming from. Where's the spillover source? And the reason why this is so difficult to do is because we are talking about a landmass in which the US, China, and India can all fit comfortably. So this is Africa, and I'm cheating a little bit because there's not that many animals up in the Sahara, but the Congo Basin is that large square, and that small square is where that 2014-2015 outbreak happened. So the previous outbreaks all happened in the big square, and that's a massive area, and that area has an incredible amount of biodiversity. So you can imagine hiking around, like casting a net over this wide area and hoping to get lucky. needle in a haystack situation, except your haystack is seasonal, it's moving, there's millions of hay pieces on top of each other, they're all interacting with each other. It just feels impossible to do. So it's an extremely difficult problem. And the spillover dynamics are really complicated. So there's a lot of question marks on this figure, even though it was published in 2014. And you can see that there are lots of animal species that have been implicated, and it's mostly because these species have been found dead, and then when you sample them and try to figure out how they died, they're teeming with Ebola virus. So great apes are actually under threat of extinction. If we do not get a hold of the Ebola problem, great apes will go extinct. That's what all of the mathematical models are showing. There are forest antelopes called duiker, red river hogs that are a suspected reservoir, rodent species. So there's all kinds of pathways that are still to be discovered. And of course, the main hosts are these bat hosts. So here are all of the outbreaks that have happened. And the filoviruses that are in green on the screen are the ones that are responsible for the biggest human outbreaks. And among those three, Zaire is the one that have caused the biggest outbreaks. So back in 2014, we had just finished a paper that showed that machine learning could be applied to make some predictions that we think would be useful for predicting zoonotic disease hosts. And we thought, okay, well, maybe there will be enough data in the filovirus system to make some predictions there. And so the first thing we wanted to do is ask ourselves, like, okay, well, how many bat species have ever tested positive for any filovirus ever? And those of you who are scientists in the room know that there's a difference between finding active, like live virus in an animal or even genetic material, like by PCR, in an animal, and it being seropositive, which just means that it's showing some antibody response to having been exposed to Ebola virus at some point. It might not even be actively replicating anything, but it did get exposed at some point. So it's showing a signal of Ebola in the environment. So at the time, there was no maps. There are no maps. So we put together this map just to figure out where the ranges were of those 21 species that were known at the time of being positive for any kind of a filovirus, okay, even the ones that don't infect humans. And when you look at this map, that star is in a red zone where the last outbreak happened. So it makes me ask myself, what if we had had this map before the outbreak? What would we have done differently? And that question is still not clear to me because our predictive ability is still pretty coarse. Okay, so let's zoom in a little bit. If we zoom into this area, we can see what the problem is. So we see that red area, Guinea, Sierra Leone, that area that's glowing red. Ghana is also bright red. We've never had an outbreak in Ghana. That's very interesting. One would ask oneself why. And then Uganda, Rwanda, all that area, that is sort of like Ebola zone. We know that Ebola breaks out there. Okay, so the task here for prediction is, we know what the pathogen is, it's any filovirus, and we don't know what the bat species are that we think are capable of being a reservoir for this pathogen, and we don't know where they're gonna be. So how did we attack this problem? Well, we reasoned to ourselves that the things that we do know about these animals that distinguish them from each other are represented by this figure here. So this isn't all of the features that we're interested in, but if you look at an animal, you can kind of tell what it's doing in the environment by its shape, by the size of its ears, the size of its eyes, whether you're looking at it scurrying around at night or in the daytime, whether it's found with large nests full of babies, or if it's solitary, or if it lives in groups. So all of these features are quite important because they represent a picture. They give you a picture of a successful strategy that that animal has employed over evolutionary time to deal with tons of selection pressures, including parasites and pathogens. Okay, so this statement that species traits reflect ecological function and their evolutionary history is something that ecologists have known for a long time, and it's something that it's sort of our bread and butter, right? So scientists study features like these to help us understand the differences between animals and how they're living their lives in the world. And so we asked ourselves for this first paper, can traits predict zoonotic reservoirs? And the first instance in which we applied machine learning to answer this question was in rodents. And we knew that it worked because we just had this paper published. So hot off the press, we thought, okay, well, the method seemed to work for rodent species. It was able to predict which rodent species are likely to carry new zoonoses that we haven't yet surveilled them for. Maybe it'll be successful at doing the same thing for bats and Ebola virus. Let's see if it'll work. Okay, so the punchline is that it did work. We published this paper back in 2016, so it's quite old. But I would argue that there's still a lot of work to be done here. I'm going to just walk you through the results really quickly, and then I'm going to circle back to how we're advancing that, on that result today. So here are some of those traits I was mentioning before. These traits capture in evolution a snapshot of how these species are existing currently, how they're functioning in their current ecosystems. And these features are easier to observe because we've had decades of basic science that underpin these data. So this is like probably armies of graduate students, PhDs, that are represented in these features. I mean, these are students that are going out, or researchers that are going out capturing the bats just for pure curiosity, trying to understand, like, what is this one species doing out here? How is it living? What is it eating? What's its metabolic load? Like, all of these very basic questions that none of them would have ever guessed would contribute to a model to predict filovirus outbreaks ever in their lives when they were collecting these data. But these are exactly the kinds of data that machine learning models require in order to make the predictions. And so when I present this work, I'm really sort of thanking all of the basic scientists that went before me that made these data available for the rest of us. So in our model, what we did was, you guys are all familiar with machine learning, whether you know it or not. I know that all of you guys are familiar with ChatGPT. This was like before GPT was cool, or machine learning was like even a thing that people knew about. So it was actually kind of difficult to convince people that this method would work, believe it or not. But the method is very simple. You guys are probably all familiar with Amazon. It's an algorithm that helps you to figure out what you want. And you can type in things and it'll surface a couple of search results, and you click around, and then eventually you kind of narrow down on the thing that you're looking for. And the way that it does that is that it learns the features of the things that you're interested in, right? Sometimes it surfaces things that you didn't even know you needed until you saw it, and you're like, yeah, now I need that thing. These algorithms are actually quite good at getting an accurate read on what you are training it to do. It's the same exact thing with this model. So this is an ensemble boosted regression model for those of you who are interested. It's a vanilla, straight off the shelf. I did not develop this model. It's been known to computer scientists for years. It was just really well suited to this problem. And what it does is I tell it which species are currently known to be reservoirs of or carriers or seropositive for Ebola virus or filoviruses. Okay, so you have a 1, and then all the other species are zeros. So you're training the model to learn the features, these features of the 1s. So learn what the 1s look like. Learn all of the features that characterize a 1. Learn the relationships among those features. And once you've got that learned, I'm going to test you on a data set that I've held out and locked up to test how well you do at that prediction. And once I've decided that the training is done and the test result is good enough, I set it loose on all of the data. And what it gives me is a prediction of what a good carrier looks like and which species are likely to be carriers that are still zeros because we haven't been successful at surveilling them yet. So here's what we found. I'm just showing you here the top 90th percentile of probability of the bat species that the model predicted to be carriers of Ebola virus. And you can see some interesting patterns here. You can see that there's a lot of activity in Southeast Asia. We'll get to that in a second. There's obviously a smear across Africa. There's a bunch of species there. And there's a lot of, unsettlingly, there's some activity in North America and South America, which we kind of scratched our heads and thought about for a while, but then it turns out that the filovirus evolutionary biologists are very excited about this result because they have found in the Myotis group, which is a very common species of bat in the Americas, that there are parts of the filovirus genome that are in the bat genome, which is fascinating to think about. How did it get there? There are no Myotis species in the Eastern Hemisphere. They're totally neotropical or North American bat species, and they have these remnants of the filovirus genome in their genome. So that's a very interesting thing to follow up, another hypothesis for another day. But if we zoom in on this area, we have had some species that our model predicted that have since been confirmed, but confirmed as positive for a new filovirus. So this Bombali virus is a new filovirus that was confirmed to have the ability to enter human cells, Bombali virus, and it was found in these two species, which our model predicted were pretty high up on the probability list. And the scientist who was involved in the team that discovered it, his name is Simon Anthony, he emailed me like immediately. I was like, Barbara, we just found Bobali virus in these species and they're on your list. Maybe we should just keep going down your list. And I was like, that's great, I think. I mean, you never want to be right about these things, but it's nice to have a guide, right? So we had some confirmations early on that were important. If you click in on Southeast Asia, here's what we see. There are up to 26 bat species that are overlapping in space and time in this region of the world. And we've had outbreaks in all of these countries since, or not outbreaks, these are all of the countries that have had research done in them to suggest that there should be the capacity for many species to carry these filoviruses. And yet we have not had a massive outbreak in any of these places, which is very interesting to think about why that is. Maybe it's evolutionary, maybe there's some background immunity, maybe the ecology is different. Anyway, lots of hypotheses that are being surfaced just from this one result. Eococcyx is displayed here because it was number 5 on our list. It was not studied. We didn't know if it carried filoviruses or not, but it was pretty high up there. I think 6 months after our paper came out, there were other papers that came out that confirmed that not only was this species positive for filoviruses, but it was a new tropism, which means that the pathogen was found causing infection in the lungs in a different part of the body than ordinarily found in bats. So being in the lungs is not great if you're a pathogen that could spill over to humans. It indicates that it could be aerosolized. So we had some successes from this approach. And this is an approach for which there was at least some data, right? It's not great data. It's 22 species out of 1,500 bats now. So very small sample size. Also, the predictions are really coarse. I mean, what do you do with a prediction like that? Let's just cast a net over Southeast Asia and look for these 26 species. That doesn't seem like an actionable outcome. It doesn't actually help you make management decisions, right? So, this was published many years ago now. We're scratching our heads. We're seeing new outbreaks happen. It's not clear what to do about it. What do you do when you have even less data than that? So that's the situation we found ourselves in when COVID happened. So as zoonotic disease ecologists, we watched COVID spread around the world and infect all these people. And we knew that the evidence suggested it had come from a bat species or some animal species in one region of China. And we thought, okay, well, it came from an animal, and now everybody has it, and now it's going to get back into animals. And then there's going to be this weird back and forth, and what does that mean for the future? So, but there wasn't any information, right? Like, we didn't have a bunch of positives that we could train a model on. We didn't even know what we were dealing with. And so we just kind of got stressed about it for like a month. Like, we were all very stressed, I'm sure. My lab and I kind of started meeting remotely and just wishing that we had something we could do, because it felt like exactly the kind of thing that our group should have been able to predict. But how do you predict when you have no information? And so this was the team whose work I'm going to present. And you'll notice we have Arvind Varsani here, one of your very own. I'm very proud to have worked with him. Adrian and Ilya were postdocs in my group. Adrian's still there. And Joao Rodriguez was a cold call. We found him on Twitter because he had published a study, and I thought, oh, you do structural biology. I think we need to talk to you. So it was really Ilya who I credit with making the statement, like, Barbara, I know it doesn't feel like we can do anything, but man, it sure feels like we should try. And I looked at him and I was like, you're absolutely right. We just need to dig in and see what we can do. And so that's what we did. We started working I was homeschooling my kids, and so that was not a fun time, but I stayed up very late at night, 2, 3 in the morning every night, reading these papers that felt like they were coming out like drinking water out of a fire hydrant. It was just so rapid. And we tried to figure out how viruses bind to cells and what can you do with that information, and how come nobody has any information on coronaviruses except for people here who had been studying it before it was cool, and before the science was needed. that's why we had the information that we did have at the time, because there were scientists who were studying coronaviruses for the sake of understanding coronaviruses. And that kind of basic science is why we are persisting as a species and our lifespans are longer. So we asked ourselves this very fundamental question: what do viruses need to get into humans? Well, they need a receptor. They need some way to get in. They don't always need a receptor, but in this case, they really like to use this ACE2 receptor in humans, which is very important for basic metabolic function, like controlling your blood pressure. So it turns out that ACE2 is actually highly conserved across all vertebrates. So all vertebrates have some version of ACE2. They look a little bit differently, but they all have it. And so what happens is this virus binds to the ACE2, it gets into the cell. So we asked ourselves, okay, maybe we can leverage the molecular structure to tell us something about this virus and tell us something about what host species this virus should be able to bind to. And our logic went something like this. Structure in biology always tells you something about function. The way that something is shaped, even if you're talking about a whole organism, right? If you're talking about a molecule, it's even more important. All of these little binding points are really important for how the biochemistry works. If it's not going to lock in at these points, it's not going to be able to get into the cell. If a drug isn't shaped exactly the right way, it's not going to give you the properties that you want it to. And so structure absolutely underpins function. So we thought to ourselves, okay, well, if structure underpins function, and all of these species have ACE2, and the ACE2 structure is a little bit different for each species, that difference must be meaningful. And that difference must propagate through all of the other traits that we do have access to. So what if we could train a machine learning algorithm to recognize the characteristics that we have data on to predict binding strength? Because we had that data. So that's what we did. We were like, oh, let's just try it. We don't know how to do that binding analysis, so we got Joao to help us out. So what we did was we took the structure-function relationship, took the host-receptor structure, took the crystal structure of the original variant, the Wuhan 1 strain, and we did the binding predictions for all of these ACE2s, for every animal that we could find. So this is what this figure looks like. The strong binding is going up, and that gray box, that line is drawn at the cat, because cats have the weakest binding that still allows for the cat to shed virus. So we thought, okay, well, if it binds more strong than a cat, then maybe that's when we know we have a problem, right? Now, I am glossing over a lot of important details in virology, which is that binding is not the only thing that matters. In fact, you can bind strongly and actually just totally fail as a virus to infect, to get packaged up, to egress from the cell, to get out of the body, and then to infect another ant. So all these things have to happen downstream. We just had information on this, and so that's what we ran with. Okay, so we have lots of different classes of vertebrates represented in different colors. We have the blue dot representing ray-finned fishes, which I'm still puzzling over because you can see the binding is quite strong for coronaviruses. Amphibians, birds, cartilaginous fishes, mammals, and reptiles. So there's lots of pink in there, and we thought, okay, well, we can't work with all of these animals. We're just going to extract the mammals and just do our prediction on those guys. Okay, so here's what the process kind of looked like. I'm going to track with me from the top right here where it says structural modeling. We first take the receptor and the virus, and we fit them together, and we measure how strongly they bind. That's what that haddock means, the haddock model. We take that binding strength, we combine it with the host data for each of those binding strengths, and then we apply another machine learning algorithm to learn what predicts binding. We have something that we call zoonotic capacity, so that's a prediction on the probability that any given mammal host will become infectious for SARS-CoV-2. And then we have our predicted host list. We have predicted host list, and we have the ones that we've been observing in the field just because people keep observing new species cropping up in the field or even dying from infection. And then we're sort of matching with field evidence, lab evidence, and ideally the circle would keep going round and round. We keep iterating, the model makes predictions, you go out and test them, and then you kind of start the cycle again. I cherry-picked some results to walk us through today, just to demonstrate a couple of things. The model did a remarkably good job. It got the known positives at the time correct. Mountain lions, great apes, and, well, the mink. Our model predicted the mink And then the mink was confirmed in the news. And then we're like, oh, we just were about to submit this paper for publication. Now we have to redo the model because it's already out of date. It just felt like I was on a hamster wheel. So we redid the model, included the mink. The species that were subsequently confirmed after our paper was published, the most surprising one was the white-tailed deer. It is so widespread, and in some countries it's actually farmed. So the white-tailed deer, obviously you know that it can reach quite high prevalence. It seems to kind of die out after a while. You still have a lot of seropositivity in the population, but there doesn't seem to be like this huge active infection happening, at least that we can detect. This is a deer mouse that was confirmed positive, although I was recently told that they have now split this into 4 different species because they're genetically different from each other. So this is like a treadmill. It's like you got to kind of keep up on the literature here. So at the time, this Paramiscus maniculatus was confirmed positive, and actually they can give it to each other when they're separated in a cage. So it's airborne. They give it to each other through the air. Red fox, raccoon dogs, and the snow leopard was near the top of our list, and a few months after our paper was published, snow leopards died in the Lincoln Zoo, which is very sad. And we ask ourselves, like, well, what should we have done with these results to prevent that? It's unclear. We can publish them. We can take all of the press releases and take all the interviews and say conservationists should be aware, but at the end of the day, it just doesn't feel like it's enough. Some of the confirmed negatives that I was happy to find out about, that is me holding a white-footed mouse that I caught on my kitchen counter. And I would not recommend being at that distance from a mouse that could have been a host for COVID during the COVID pandemic, but I'm happy to report that the latest results suggest that white-footed mice do not get infected by SARS-CoV-2. The field work that's been done shows incredibly low seropositivity, meaning they're probably not even getting exposed to it in the wild. And so at this point, it doesn't seem like it's a high risk. I have pigs circled here because our model and a bunch of other models predicted that pigs should be susceptible to COVID, SARS-CoV-2. All of us have been proven wrong. It's not clear why. They just don't get infected. And I think that this is an important demonstration that the negatives are really important. Why is it negative is its own hypothesis that we should be chasing, because what does that mean for how we think about infection for us, right? Pigs are an important source of xenotransplantation research, and so we need to keep an eye on whether pigs continue to be resistant to all of the variants of COVID, so this is not game over, but it's very interesting to think about why pigs are resistant to infection. Cattle are also confirmed negative, although it kind of depends on which variant. So the variants are a problem in the future. And this is sort of where we're at right now. I don't have a slide to describe this, so I'll just tell you that the next step in this research is currently ongoing in my group. We have a collaboration with IBM who are doing this sort of protein-protein interaction modeling for strains of SARS-CoV-2 that we predict will be zoonotic. So we're generating sequences that we think the receptor binding domain has the capacity to get into human cells. We have a sequence library that we're generating. Then you take the 3D structure and you match it up with the ACE2s to see which ones have a good fit. And then you take that into a pseudotype virus situation. You design these fake viruses that are not infectious. They just express the sort of binding partner, and you see whether it gets into the cells of interest in human cells, most importantly. But then we have a bunch of ACE2s that we're generating for the northeastern forest community, like white-tailed deer and fox and mice, because we know that these variants are able to get into animals differently depending on which animal. So if you're a predator and you're infected with a couple of variants and you're a host but you're not infected at all, or mice and you're not infected at all, what does that mean for the ecological interaction? Are the predators gonna die and then mice will go up? And then what happens after that? Like, are we gonna get a huge Lyme disease outbreak? So there are all of these knock-on effects that ecologists are thinking about all the time because this isn't happening in a vacuum, right? This is happening in real life, in my backyard, or on my kitchen counter. So these are the kinds of questions we're pursuing next in collaboration with many folks. Okay, so I'm gonna bring it back around to filoviruses now. I told you that the last set of predictions was really difficult to enact, to take action on. And that's because there's tons of bat species in the world. Bat biologists will argue about whether it's 1,400 or 1,500 bat species in the world. There's a lot of them. There are 300 species just in Africa. And there are over 400 in Asia. So there's a lot of species in areas where we know that these viruses exist. There's a bunch of different viruses. Only a couple of them have caused human outbreaks. So there's this huge combinatorial problem, right? So we have a bunch of filoviruses. We have host receptors that are all different. So like, are you going to do the matching for all of them? And we don't even have the sequences for all of the receptors. In this case, it's called MPC1. This receptor controls cholesterol. And so things like your cell membrane and whether you're more fluid or less, like more structured, that MPC1 controls that. So we're kind of thinking about like, how do you get the dual precision that is necessary to take action? Which bat, which filovirus, and is the filovirus one that I care about for people? Okay, so that is how we're trying to advance this prediction frontier. Let's go from unknown, unknown, known to like which species, where. So that's how we're ratcheting up next. And in order to do that, we're borrowing data from a group of collaborators that actually took all of the known sequences of MPC1, expressed them in the lab, did the binding analysis to see which ones are infectable by which filoviruses, generated all the curves, and they have binding information. They were really interested in the physicochemistry, so what are the actual physical and chemical properties of the binding that allow for tight binding versus weak binding. It's an incredible paper. I would encourage you to check it out. So we're taking those models and we're doing the thing that I told you before. We're asking the model to learn the features of good binding based on these experimental data, based on traits of these bats, to make predictions about which ones should carry which filovirus. I'm just showing you these two results from one model on one filovirus, which is Ebola Zaire, the one that we care most about right now. And Mops condylarus and Hypsignathus monstrosus are two that are predicted with high probability to carry Ebola Zaire. Mops condylarus has not yet been shown to carry Ebola Zaire in the wild. There are some suggestions that it's conducive to infection, so it's definitely something to keep an eye on. Mops condylarus is also, their range is, I'll show you a map in a minute, but its range is expanding, actually, in Eastern Africa. It's really great at living with humans. Hypsignathus monstrosus, I believe now there's PCR positivity for Ebola. So the model's doing a reasonably good job at making predictions that we can now take action on. So I want to leave with this point that prediction success really hinges on basic science. So we don't have complete data for all of these species. And in other analyses that I haven't shown you today, actually our models fail when we lack too much basic information. And oftentimes we're lacking basic information for species that are actually really common. Because it turns out that people don't really care that much about studying the species that are like not charismatic, not endangered, in their backyard, kind of annoying. Like we don't have a lot of basic information about what they're doing at night, where they overwinter, and all of these basic things. So I want to leave you with that point. And also this demonstration of like the range that's known for Mops condylarus is in purple. The human outbreaks for Ebola Zaire are in white. And there is no overlap. But it's not because the bat isn't there, because the yellow points are where scientists have observed this species in the wild, in the field. So this purple map needs to be updated. Has it been updated? No. Why? Because there isn't as much support for that kind of basic science, and it's very hard to do. So I don't have to convince this crowd that basic science is important, but who wants to update these maps? There are people who are not really hinging on filovirus work. They're not really interested in the viruses that these guys carry. They're interested in just understanding the biodiversity around them. And that's why our models work, and that's what prediction is going to depend on in the future. So I want to end on a positive note. I think that places like the Biodesign Institute, anytime we get to do interdisciplinary science, that's where the problems of the future are going to look to. We're never going to run out of infectious disease problems, especially with the climate issues that we have before us and with the sort of downgrading of science that we see. These problems are going to not go away because we're not looking. They're going to just be there and increase in the future. And so I would encourage any scientist who is looking at the situation right now with bleakness, hang in there, because we need all of the bright minds in this room and around the world to just stick with it. And I would also encourage those of you who have a perspective on a system that's like different. Like you see something that not everyone sees. And you look around and you're like, well, nobody's looking at it that way. And maybe you're junior and you don't have the confidence to think like, oh, my idea means something. Hang on to that idea. I think this school is doing an amazing job with diversity. And the diversity of perspectives and approaches in science is what is going to keep us afloat and what is going to push forward our prediction frontier and what's going to push forward humanity. So thank you so much for your attention. I'm really optimistic about the future, despite the fact that we have lots of diseases to deal with, because I've been around people like you all day, and I'm just very grateful. Thank you.
Wait for the microphone so everyone can hear your question.
Now that everybody has a cell phone with GPS and they can be tagged, this just opened up a whole new, or why are we not using more citizen science with everybody's cell phone? They can track all these animals in real time and tag them, and it's incredibly cheap. It costs almost nothing. They can help promote this basic level of understanding of range and different species. Thanks.
Yeah, that's a great question. We are, the collective we, are making use of citizen science data. So there's things like iNaturalist, which maybe some of you are familiar with, that allow you to take a picture, and experts will look at the picture and score it as like research grade, like how confident are you that it's that species in that spot. If it's like some species that's supposed to be in California and you find it in New York, like that's not research grade. That was you making a mistake. But there's tons of those exact kinds of data that not just give us an idea about their range, but also their timing, their phenology. So when do you see more of them? When do you see less of them? Do you see them in the flowering trees feeding on nectar, or do you not? Or are you seeing them in weird trees? So there's a lot of those data. They're difficult to deal with in their own way, but that's exactly the kind of sort of input that we need to make these models work better. Yeah.
So first, I'd like to thank you for a wonderful talk. But I have a question about the white-tailed deer. When I first read that article, I thought, oh, well, it must have been somebody, because you see these pictures of people in Michigan where the deer come into their yard and you can feed a deer with your hand. I thought,
my neighbor does that.
That's an easy one. Somebody had COVID, they're coughing into their hand and then they gave it to the deer. Is that how it happened, or?
I don't know how it happened, but I'm thinking exactly like you're thinking, because if you Google something like deer habituation or deer rehabilitation, it's incredible the pictures that will come up. Like people snuggling deer like they're Bambi, and people's like minivans like in the back filled with fawns that they found, just taking them to a rehab place. So I think that the human to animal transmission route is not that far-fetched. It's very interesting to think about the deer-to-deer transmission. Of course, they're very social, and so they don't wear masks or distance or anything. So you can imagine it racing through the population. So I don't think that the spillovers, the transmission routes, are the thing that's curious there. I am curious about why it seems to infect everything and then go away. And when you look for signals of SARS-CoV-2 now, it's actually kind of hard to find it. So like, what's changed about the deer? Is it the variants that are circulating right now? Were some variants were persistent in deer and not evolving? Other ones never got into the deer, and some of them are just, they're just not getting into the deer. What does that dynamic landscape look like, and how should we prepare for that is the thing that is very curious to me. The answer's not clear. Yeah.
Maybe I'll follow up a little on that. I don't know if you want to comment. There's been a lot of discussion about how the spread of humanity is impacting, ecological life zones of animals and various species, and how that's causing more interaction or less. I don't know how much of that is playing a role in the spread of these things too.
Yeah, I think that's a very important question, and you touched on a lot there. So one of the fastest growing kinds of urbanization is in the form of informal settlements. So these are places where lots of people are living without a lot of infrastructure. There's not services provided. They're often marginalized, ignored. They're not included in COVID responses. And so a lot of these are in like developing Africa and places like that. So these informal settlements have their own ecologies. And actually the microbial communities in these places, I think, are probably either all very similar across very distant informal settlements, or they're pretty different from each other, and you can analyze the communities to see what you think is going to emerge because of the kind of settlement that it is. And then depending on where the settlement is, if you're in Southeast Asia versus Uganda, the kinds of wildlife that are able to persist in your informal settlement are also different. So I think there's a lot going on there. There's also like a lot of really cool urban disease ecology happening in the United States and other places where there's more research funding, and we're interested in things like I have a colleague at Cary Institute, Shannon Ledoux, who's been studying mosquito-borne diseases for a while, and really interested in these sort of abandoned places in cities like Baltimore, where you have like blocks that are dilapidated and there's trees growing out of these foundations that are never gonna be torn down, tires and things people use them as a dump site, and so there's like toilets and tires and stuff, and there's tons of mosquitoes growing in those places. What is the urban ecology of the rodents that are there? Do raccoons visit those places? So there's all kinds of biology and ecology that is perpetuated by dilapidated urban systems, perfectly functioning urban systems, and then also these informal settlements that are just kind of a hodgepodge. So there's a lot to dig into.
Hi. So that last comment just made me wonder if how much you guys have thought about. I mean, you're talking about tracking connections between different kind of animals, and is there a way to maybe tag that by adding a known compound that you could then trace to which other kinds of species and locations that it goes to, like a radioactive tracer kind of thing?
Like a contact network to develop. Do you mean like developing a contact network so that you can see which animals are contacting which?
Right.
Yeah.
Have a tracer where you could see you could, just like you do in the body, you put in a radioactive tracer and you see where it ends up. And could you see whether, this tracer ends up in mosquitoes and then it ends up in different kinds of animals and plants? Where does it end up, but that you could actually track it some way?
I'm going to say yes, but I don't have a lot of details on how it works. Like heavy carbon, heavy hydrogen, I think, is one of the tracers that people use pretty often. Stable isotopes is another way that people have traced the movement of energy materials through a system. In terms of using it as a tracer for like contact, there are various ways that ecologists have done it. One way is to set up like a grid where you can have like RFID sensors that are in the rodents, and then every time they pass by one, it sets off a ping. And when they contact each other, it sets off a ping. So you can kind of reconstruct the contact network and also their movement patterns for individuals if you have a grid set up like that. So I mean, it's very dependent on the research question you're asking, but that's one way. I don't know of any sort of like reliable, large-scale contact tracing between multiple species in an ecosystem. But I think that would be really cool. I don't know of any at that scale.
I think there was one back there.
I know that you started this work quite a while ago when the machine learning landscape was rather different than it is now, but it seems as though you've stayed with the boosted regression models throughout that time. I was just wondering if you've tried neural networks or other models and if they haven't worked, or if you've decided not to try them for some reason.
Yeah, we did think about using other methods. We found that they each had their issues. Boosted regression seemed to be the most stable performing. And so we stuck with it for that reason. Like neural nets are a little bit prone to overfitting. And then others are really dependent on like your initial conditions. So there were some technical reasons why, but we also didn't like do an exhaustive comparison across all of the different ways to do an ensemble model. Like you could use boosted regression, you could use random forest. Random forest anecdotally does better on wide data sets where you have like lots of features in shallow rows. We had the opposite, where we had many fewer features and lots of rows. And boosted regression seems to do a little better there. I mean, it's a bit of an art, even though it's quite technical. So yeah, that's the short answer. It's like kind of, we checked, but boosted regression seemed to work, so we stuck with it.
Thank you for the great talk. This question is being asked by my neighbors and my friends, whether SARS-CoV-2 is synthetic or is natural. But I don't have enough knowledge to tell them the answer and ask some people. People have different views. So based on the current literature and the findings in the lab and the theory, so what do you think?
I think it's natural. Yeah.
Why is that?
I'm also not a virologist. Actually, there are experts in the room who study coronaviruses, like the experts in coronaviruses, that we could ask for more detailed answers. But just based on my understanding of the literature, of the peer-reviewed literature, I think most evidence, all of the evidence points to the fact that it's a natural pathogen.
There's one in the back, I think.
Yeah. Thank you so much for a fantastic talk, really. I'm just a little curious, initially when you're talking about the three groups of having two unknowns, which one do you think would you rank in terms of the difficulty in their approach? Or I guess, which poses more risk? And what would you do, what kind of steps would one take to kind of level the playing field among the three groups of having two unknowns?
Yeah, so you're asking really about like prioritization.
Yeah, kind of.
Yeah, boy, that's tough. I think in reality, you do what you can, I think is the real answer. If you know that a place is a problem, then you just go and see what you can learn about the place. But if you're in the middle of a pandemic, you just zero in on the pathogen, right? And you try to figure out the rest of the unknowns. So I think that's a really insightful question, and I don't know how I would prioritize it, but I think in reality, it's just you do what you can. Yeah.
Hi. So you said you started on amphibians, right? So do you have any insights, or what are your thoughts on aquatic and maybe marine, I lean more marine, diseases, and even maybe what might be coming out of ice melt?
Oh, yeah. Well, I think the aquatic systems are understudied. I think marine systems are very understudied when it comes to infectious diseases. It's just more difficult to study. I'm concerned about the urgent nature of climate and warming waters in general for biota overall. And so I think the pace of change in the oceans and in water is fast. I don't know if it's faster, but it feels more looming to me because the consequences feel like they're about the scale of the coastline. So it's like a huge amount of biota and interactions that are being affected all at once. I forgot the first part of your question, Lisa. You're asking me about...
Yes, so since you have experience in aquatic systems, I was wondering if where you want to, if you are at all going to look in that direction again.
Yeah.
Because if you look at marine diseases, no one is, I don't think, doing any prediction work, which is hugely needed. There's a lot of situations where it's not viruses but bacteria, and it's not just one bacteria, it's consortia. So these things are really hard, and there's a huge outbreak across the Caribbean that started in Florida, and it's spreading all the way to South America. So that's not even linked to ice melt. This was through the dredging of the Miami port. So there's a lot of work there if you're ever bored.
Thank you. Yeah, I think the aquatic interface is really interesting, the freshwater interface specifically. Because there's a lot of ecosystem linkages between water and land that I think are not connected because of the disciplinary divides. So one example is recently a colleague of mine at Cary Institute, who's recently retired actually, Emma Rosie. She's an expert in pharmaceuticals in our waters, essentially. So illicit drugs, prescription drugs, and things like that. And one of the things that she found is that some of these aquatic insects that have one life stage in water and then emerge as adults out of the water bioconcentrate the active ingredients in some of these pharmaceutical products. So what happens is you bioconcentrate it, so it's diluted, but you're concentrating it, and then you get eaten by a predator, and then it's more concentrated, and then on and on. So she approached me and said, Barbara, there's a lot of antivirals that are being administered right now, not only for SARS-CoV-2, but for seasonal influenza, which can sometimes be quite severe. So there's more antivirals available. And I know from my own work that our wastewater plants are not designed to remove the active compounds. And in fact, there's some research to show that if you're sampling water kind of closer and closer to a pharmaceutical production plant, the concentration goes way up. So she posed the question, like, what does that mean that we have all of these antivirals in our water and our aquatic insects bioconcentrating them? And then when those insects emerge as adults, what eats the insects? Bats. Bats actually specialize in those insects. And in fact, there's like feeding frenzies over emerged insects in some of these places. So those linkages between aquatic systems and vertebrate systems on land and humans, very understudied. Definitely some things are happening. It's an area of research that I would like to pursue, but yeah. If anyone in here is interested in a PhD.
Oh, hi, hello. Thanks for your talk. I have a question about the data. So I would like to know if you include the microbiome abundance data in your model.
No, we did not include microbiome abundance data. I think that's generally unavailable for most animals, unfortunately.
And what about any other like genetic sequence data?
Genetic sequence data, not for hosts. No. Because the data that we use to train the models for hosts are averages, central tendencies at the species level. So they're not individual, they don't represent individuals at all. So, okay. But I think that you're correct in that there should be sequence variation among populations of hosts, and that sequence variation actually is quite important for helping us to zero in further, because populations, of course, are different from each other. Even if you're calling them the same species, they're different. They have different ecologies, and actually their behaviors could be quite different. So yeah, that's another way that we could kind of ratchet a little bit up, I guess.
Kind of related to that, I was wondering about immune systems.
Yeah.
It's probably too hard to capture that, but is there a way, as you look at the host, to categorize their immune systems?
Maybe in a coarse way, but actually there too, I think that there's so little data on what the important axes are for different host groups that it's just a wide open area.
Okay, well, let's once again thank Barbara Hahn for a great
presentation. Thank you.